In this video, let's discuss the topic apoptosis. Apoptosis means programmed cell death under genetic control. Examples include death of neurons in CNS. It may be physiological or pathological, and degeneration of fetal tissues, that is, like webbing of fingers and eosinophils. Mechanism of apoptosis. There are some enzymes called cysteine proteases, commonly called as caspases, that trigger apoptosis. In inactivated stage, they are present in normal cells. When they get activated, they cause apoptosis. There may be two pathways for their activation, that is internal stimuli or external stimuli. Internal stimuli. Internal stimuli is due to intracellular stimuli such as apoptosis inducing factor present in membranous mitochondrial space that reaches nuclei and causes destruction of DNA, further results in activation of caspase 9. External stimuli such as first apoptotic signal is produced by tumor necrosis factor, which binds to FAS ligand present on target cells that results in activation of caspase 8. Activated caspase 8 and 9 are called as initiator caspases. They further result in activation of executional caspases such as 3 and 6. Further results in proteolysis, which results in hydrolysis of sphingomyelin present on cell membrane that causes ceramide generation, which causes mitochondrial injury. Further DNA damage occurs by reactive oxygen species and free oxygen radicals produced in these reactions causes release of nuclear protein TP53, which induces synthesis of BAX. BAX is nothing but the cell death promoting protein that results in cytoplasm and chromatin condensation and formation of blebs on cell. These are the blebs present on the cell, which further forms apoptotic bodies and then apoptosis, further phagocytosis. Here, apoptotic bodies bring macrophages by producing thrombospondin and phosphatidylserine on their surface. Normal cell undergoes shrinkage that further forms blebs on their surface, which results in suppression called as apoptotic bodies. These apoptotic bodies has thrombospondin and phosphatidylserine on their surface that attracts the macrophages and further results in engulfment by macrophages, that is known as phagocytosis. Regulation of apoptosis. There are certain genes such as pro-apoptotic genes and anti-apoptotic genes. Pro-apoptotic genes promote apoptosis and anti-apoptotic genes inhibit apoptosis. They include BCL1 to BCL-XL, MCL, and anti-apoptotic genes include BX, BAK, etc. Physiological reasons for apoptosis, that is endometrial shedding during menstruation, regression of breast after cessation of breastfeeding in lactating women, normal shedding of intestinal epithelium, and degeneration of thymus with age. Pathological reasons of apoptosis include tumor cells death during chemotherapy, transplant cell death in resection of the graft, pathologic atrophy, that is after orchiotomy prostate atrophy occurs, degenerative diseases, that is Alzheimer's and Parkinson diseases, cell death due to radiation, hypoxia, and thermal injury. Most common characteristic features of apoptosis is the inflammatory response is totally absent in apoptosis, and cytoplasm is reduced, cell organelles are normal, chromatin condensation around periphery of nucleus occurs, and phagocytosis of apoptotic bodies by macrophages. Let's see few differences between necrosis and apoptosis. Necrosis is a pathological cell death which has the main three processes: pycnosis, karyolysis, and karyorrhexis. Pycnosis is nothing but condensation and clumping of nucleus. Karyolysis is disintegration of nucleus, and karyorrhexis is nuclear fragments get integrated into the cytoplasm. But in apoptosis, only pycnosis step is present.